afternoon. Good afternoon, everyone. So the title of my presentation is Food Effects on the Gastrointestinal Transit Properties of Amphotericin B, prepared in solid lipid nanoparticles. We've heard a lot about nanoparticles this morning, and particularly about amphotericin B. I was quite fascinated on the talk just before lunch about amphotericin B. So what we've done here is to look at the food effects on the gastrointestinal transit properties of amphotericin B. But before I go into the details of the work, just a brief introduction that this drug is very effective, as we've heard this morning, in treating very serious fungal infections. However, it's currently given by intravenous route, and it comes with very serious side effects, as we've come to know. And research has shown that this drug can have slightly lesser side effect when it's given by the oral route. However, it's very poorly soluble, as we've heard this morning as well. So there is an interest to develop this drug into alternative delivery systems, which can improve the bioavailability. And so we are interested in this area as well. But we are aware that no work has been carried out to see the transit properties of amphotericin B dosage forms, and particularly to see which part of the gastrointestinal tract this drug is best absorbed, if the oral route is to be chosen. So the purpose of the work was to formulate amphotericin B in solid lipid nanoparticles intended for oral delivery, and to study the gastrointestinal transit properties of the dosage form, that is the solid lipid nanoparticles, but also to see where exactly in the GIT amphotericin B is best absorbed, and also to see the effect of food as well. So whether food has any effect on the absorption of amphotericin B within the gastrointestinal tract. We decided to use a very not so sophisticated method, but it's nonetheless acceptable. So we use marker drugs to study the gastric emptying properties, as well as the cecal arrival of the dosage form. So amphotericin B was prepared in the same way as paracetamol nanoparticles, and the paracetamol, as you are all aware, you might be aware, is very rapidly absorbed from the small intestine. So it can be used as a means of determining the gastric emptying, or the arrival of the dosage form in the small intestine. Sulfasalazine can be hydrolyzed into sulfapyridine in the large intestine and be absorbed, and therefore it can also be used as an indicator of the arrival of dosage forms containing this drug at the cecum. So these were prepared and were characterized with regard to size, charge, morphology, among other things, and were studied on animals. So we used male Sprague Dowley rats weighing between 250 grams. And of course, the work had an ethical clearance, and the animals were divided into two groups. So there was a fasted group and the fed group. And each animal was administered a single dose containing the 3 drugs in the nanoparticle delivery system that had been dispersed in deionized water, and blood samples were taken from the tail at predetermined time intervals. In terms of the gastric transit parameters, the gastric transit time, or GTT for short, of amphotericin B was measured as using the plasma concentrations of the paracetamol. And that's, as I said before, is because paracetamol is very effectively absorbed from the small intestine, and that can be used as a measure of the arrival of the nanoparticles in the small intestine, or gastric emptying. So that time was taken as a time for 10% absorption, because the nanoparticles are a dispersed system, and it's a continuous system. So the arrival is not a one event, it's a continuous event, just like pellets. So 10% absorption of paracetamol was taken as the time for arrival of the nanoparticles in the small intestine. And the time for complete emptying of the nanoparticles from the stomach was taken as a time for 90% absorption of paracetamol. That was the gastric transit time. That is the time that the solid lipid nanoparticles remained in the stomach. The small intestinal transit time, which is the time that the dosage form spends in the small intestine, was taken as a difference between the T10S, which is the time for 10% absorption of sulfapyridine in the large intestine. So that was, we subtracted the T10P, which is the time for 10% of paracetamol absorbed, from the time for 10% of sulfapyridine absorbed in the cecum. Okay, sorry for the lots of, but I need to define these parameters before I go into the details. So the cecal arrival time, I said, is the time when the dosage form appears or arrives at the cecum. And it has been measured in some research work as the time when the sulfapyridine is detected initially in the blood. But this could lead to an underestimation, sorry, it could lead to an overestimation of the time because of the fact that sometimes you have the drug being released from the dosage form, so that could be detected rather than the dosage form actually appearing or reaching the cecum. So we decided to use the cecal arrival time as the time for 10% of sulfapyridine absorbed. That we felt was much more realistic. The colon transit time was a time for 90% of sulfapyridine absorbed, or T90S. So the idea was to try and produce these nanoparticles such that they had the same or similar characteristics. So they were characterized in terms of their physical properties. And the graph on the left, the bar chart, is showing the zeta average. So these were statistically not different. So the paracetamol, amphotericin B, and sulfasalazine nanoparticles were quite similar in terms of their size. Also similar in terms of their morphology from the SEM image. They look quite similar and well dispersed. And the zeta potential were quite comparable as well and were negative. We heard this morning how negative zeta potential can be relevant. These were negative as well. So we did a drug release study, and if you can see in all the cases, the paracetamol had the highest release, in vitro release, followed by sulfasalazine and amphotericin B. This is actually expected because paracetamol is very soluble in water compared to sulfasalazine, and sulfasalazine is also well soluble in water compared to amphotericin B. The graph is actually showing the counterpart for the freshly prepared as well as a freeze-dried counterpart, and there wasn't much difference between the two pairs, as you can see. We also tried to see how the particles behave when they are subjected to some kind of constraint. So we exposed the nanoparticles to agar. That was sort of to see how the nanoparticles would behave in the presence of food. I guess there could be some other way of doing this, but this was nonetheless quite relevant. So we exposed the nanoparticles in an agar gel, and then we applied some potential to see how they will migrate when they exposed to this charge. And you can see in the right panel that the extent of traveling as well as the rates was quite similar in all three cases. We also tested the viscosity of the nanoparticles, and all of them were essentially quite similar. So this gave us the belief that we can use the profiles that we have from the pharmacokinetic parameters to give an indication of how all the nanoparticles will behave in vivo. So this is a key assumption, is that since all the particles are quite similar in terms of their physical characteristics, we believe that we could use the behavior of one of them to infer how the others will behave as well. So it's a very key assumption. We believe that is the case. These are actually graphs showing the effect of food on the absorption of paracetamol. The left graph is showing the profiles for the fasted and the fed states. And the one on the right, the one on the left is paracetamol, and the one on the right is amphotericin B. I think the key thing I want to emphasize here is that in the case of paracetamol, you can see that there is a peak right about here, no pointer. I hope you can bear with me. So there's this peak over here, which is about 8 hours in the fasted rats and paracetamol administered rats. And we see a similar peak as well in the case of the amphotericin B, also about the same time. And we believe this is because of the slow process. Since these are lipids, they are taken up by the pious patches and through the lymphatic system and empty into the blood much later. That's why this appears late, and it seems to be irrespective of the drug load. So whether it is amphotericin B or paracetamol, we find that this is happening, and especially in the fasted state. So it gives that peak much later because of the slow process, the slow uptake process of amphotericin B, and that's why that happens. In the fed cases, however, you find that the profile is quite flattened, especially in the case of amphotericin B, it's much flattened. Food tends to sort of slow the rate of absorption. Again, this is something that is expected, and something similar is observed in the paracetamol profile as well. So if we put aside the profiles for amphotericin B in the fasted state and the fed state, against the absorption from the control, which was just a suspension of pure amphotericin B in deionized water, we find that in both cases there is a significantly higher absorption from the amphotericin B compared to the suspension. So in the fastest case, that's what we see over here, and so in the fed case, again, is higher. But statistically, we observed that there was only a significant difference statistically in the case of the Cmax, as you can see over here. Yep. And the AUC was comparable in the case of the fed and fasted state. Yep. The Tmax obviously was delayed because of the presence of food. Again, this is something that is expected. If we compare between the control and the amphotericin B solid lipid nanoparticles in the fasted state and the fed state, so separately, again, we find something interesting. In the case of the amphotericin B solid lipid nanoparticles, we find that there is a delay in the Tmax with the nanoparticles compared to the control. So it's 8 hours plus in the case of the amphotericin B nanoparticles, and it's just 1.5 hours in the case of the control. And the Cmax is about twice the case of the amphotericin B nanoparticles, and it's just about 2 times again in the case of the AUC. And we see a similar result in the case of the fed state, but the fed state seems to have a slightly better profile in terms of the, if we compare with the controls, I'll just take them separately to reduce the confusion. If we compare the amphotericin B solid lipid nanoparticles in the fed state, again, we find that it's highest in the case of the nanoparticles compared to the control. Yep. So similarly to the fasted, we find the same situation over here. So we try to compare the transit times, that is the times that the dosage form spends in the different sections of the GIT, in both the fasted state and the fed state. And you can see in all the cases here, if we compare this second column with the first column in this first table over here, you find that there is an increase in the transit time in the fed state, also an increase in the small intestinal transit time in the fed state, and the same is the case with the cecal arrival time and the cecal transit time. All of them seems to be increased when there is food in the stomach. Again, this is something that is expected. Obviously, CTT is most significant in all the cases. And we try then to look at the percentage absorption in the different sections of the GIT, starting from the stomach, small intestine, and colon. First, in the fasted state, we see again that we have 44% absorbed in the intestine, just 2.8% in the stomach, and 53% in the colon. We compare that with the fed state, we find a similar profile, a slightly smaller percentage absorbed in the small intestine, but a much higher percentage absorbed in the colon in the fed state. The reason why we have this colon plus here is a suggestion that the absorption of the solid lipid nanoparticles, or the uptake of the solid lipid nanoparticles, is occurring while the dosage forms would have reached the colon, but also it's likely for some absorption to be taking place through the lymphatic system, which is a slow process anyway. So it's a combination of the uptake process while the dosage form was in the small intestine, while it goes through the lymphatic uptake, plus what would be happening in the colon itself. That's what's given by the plus. And on the basis of this, we believe that obviously this is just summarizing some of the key points, that we believe that since the food has delayed the transit of the dosage forms in the gastrointestinal tract, it appears that there is better absorption. That is to say that the uptake process is very slow, and as a result of that, its emptying into the lymph is much delayed. And therefore, we believe if we can develop delivery systems to slow down this process of transit within the GIT, we are likely to have a better absorption as well. So in conclusion, we can say that we've been able to successfully produce these nanoparticles in solid lipid form, and it's certainly much more better in terms of its bioavailability compared to the control in both the fasted and fed state. We've also seen that food doesn't seem to affect the bioavailability that significantly, and the absorption process in the small intestine appears to continue even after the dosage form has reached the colon. So this is a key point that we've observed in this study, and therefore further work to be done, or further work done on delivery systems containing amphotericin B aimed for oral delivery should be made to have a slow transit. If this transit is slow, then it's likely that the absorption will be much more enhanced. And actually, we're working on this at the moment. Thank you very much.
This is certificate of recognition.
Thank you.
Great work. Continue the good work.
Thank you.
Thank you very much. There is an announcement.
Good afternoon, ladies and gentlemen. From morning we are hearing about this nanotechnology and its very specific applications in the modern drug delivery systems. Now this presentation is somewhat different. We are considering this nanotechnology if can be used in our traditional systems, and how it can be explored and how it can help in the healthcare systems. So basically here we are discussing these traditional systems, difference between traditional and Ayurvedic systems, and how this nanotechnology can be used in these traditional systems with what are the challenges and opportunities that also will be discussed. So this traditional medicines, this consider actually the total, as per this WHO definition. It's a health practice, this approach, knowledge, and these plant medicines, mineral medicines, and including all these things. Rather, it is considered this whole human body, not a specific part or specific organ. This traditional medicines, it is available whole world. Particularly important is this traditional Chinese medicines. It's a TDM, it's very famous systems. In India, it's Ayurvedic medicines. In Arab countries, this Unani medicines and homeopathy medicines, as well as some other medicines are also available in different parts of this world. These major traditional medicines or medical systems, as you explained once again, in these different regions or the different parts of this world, it is available. I think everybody knows about this one. Now, major two systems, these Ayurvedic medicines and traditional Chinese medicines, which is most popular one in this world, it is basically same, but it may be explained in different way. See, Ayurvedic medicine is considered to be this most ancient of all medical disciplines because it's around 7,000 to 10,000 years back this Ayurvedic system has been developed. In Ayurvedic medicines, the patient is viewed as unique and normally as what is appropriate for this particular systems. This whole human body is considered here. And this is in contrast to the Western medicines, where these populations are generalized, and the normal means what is applicable to this majority. So here, this Western system, we consider the particular system, whereas in Ayurvedic systems or traditional systems, this whole human body is considered. So basically, this Chinese medicine also philosophically the based and these holistic systems of medicines, and it's a balance between this harmony, peace, and these elements. So philosophically, these Ayurvedic systems are the same with this the Indian Ayurvedic systems. Now, these traditional medicines, see here it is comprises of this the different the therapeutic systems. This is plant medicines, and this also includes the herbs or this herbal extracts, herbal preparation, and different finished products and all these things. It's a plant and plant products, and this is established widely acknowledged to be safe and effective for this purpose. So this safety and this effectiveness being a major concern for this drug delivery system, where this nanotechnology is working. Hearing is okay.
You want to use the floor, what's up and down?
It's okay. It's okay. So the this potent and effective as they were 1,000 years ago. As you know, these traditional medicines are used hundreds and hundreds or 1,000 years back. Today also same potent, but only thing it is not much explored. So this alternative wide range of this, it alleviates this wide range of these diseases. Almost is nature has given or God has given this remedy for all diseases. Hence, this call for this development of delivery systems for their this attribute and this the principles. So this is why that this traditional system, which is long before from this earliest civilizations, people are using these systems. Why not this is being much explored with this nanotechnology? So this can be more useful, particularly in perspective of this safety and effectiveness. The opportunities for these traditional medicines to challenge this the modern healthcare systems. Now here we can see that due to these advances in the medical sciences, this lifespans has been increasing, and leading to this the new set of these health challenges around age-related diseases. These age-related diseases is nowadays is a very common, like this cancer, cardiovascular diseases, diabetes, and so many diseases. So many drugs are for treatment and prevention of chronic diseases, and this requires this long-term use, and this results ultimately again this toxicity and other problems, and this may aggravate over time. So development of this drug resistance and compensatory mechanism is often an issue upon the treatment. The traditional medicines utilizes the system biological approach. These whole biological systems are used here to target these multiple mechanisms of diseases treatment, prevention, and rather than this focusing on this treatment with this single chemical medicines. Here, this comparison, the traditional medicines, it takes this total, not this single drug or this the surgery process. So to overcome these differences or the different challenges, the traditional approach can be applied. Now, see this modern world, how it is complicated. We have these different the physical factors, physical stresses, these environmental problems like the cold, heat, pressure, radiation, atmospheric problems, gives a lot of pollutions, and this mental stress as the society is developing day by day in the 21st century. We are in every moment anxietic situations and leading to different mental, social or economical problems, and this also gives these disorders. And these chemical problems, this chemical water pollutions, metal pollution, pesticides, and different type of other food pollutions. All these things cumulatively, it gives the problems or disorders in our biological systems. This leads to this mutation, enzymic level, and gene levels, and different other the free radical formations. This is the mutations, and this ultimately lead to this new syndromes of this lifestyle disorders like diabetes or cancers or many other diseases. So what do we need? We need all these factors, resulting from the modern scientific technological advances, the disturbing this universal law of nature leads to this increasing prevalence of these diseases like this diabetes, cancer, hypertension, cardiac disorders, and this needs long-term, safe, and effective therapies. Besides the improvement of this quality of the life, so this changing concept of this health and disease, and the understanding of this the life process have demonstrated that the cure of an ailment is no more the sole responsibility or possibility of a chemical drug or the knife of a surgeon. It's not this only the single drug, whatever maybe its delivery, or this surgical process. This is not enough to give this quality of the life or quality of control of the disease. So this need the horrible drug developments. Why? Because this morbidity and mortality, this is due to these drug reactions, changing of this environmental conditions, this causing the many diseases, as I told you. Changing this lifestyle leads to this increasing incidence of this lifestyle diseases, and this aging or this growing global populations require evolutions in the drug development systems. So this is our problems. In contrast, the herbal drugs, they are very inexpensive and long-term use. They are naturally available. They are very natural, eco-friendly. No adverse effects in general, and this is easily available in everywhere. Almost in every country, there are some medicinal plants. Yes, in the global situation, if you see the herbal medicines, of course, used in the different parts of this country. This is in the developing country. There are a lot of use, even this 90% or 70% populations are used. Whereas in the developed country, there are also used even in USA or Australia or France or Canada, Germany, everywhere these are used, but to some extent. Novel drug delivery systems. These novel drug delivery systems, as you know everybody, that it gives a lot of advantages. The basic goal is to provide a therapeutic amount of drug to this targeted site of the body. This is which is ultimately aim of this the nanoparticle. It maintains the desired therapeutic efficacy of the drug and minimize the levels of the side effects. So these are the major goals or aims of this the nanotechnology. These novel drug delivery systems, it has there are many hurdles are there. If these hurdles are overcome, then this herbal drug delivery can be successful one. These challenges are water insolubility of the drug. The many herbal extracts, they are not soluble, hence they are less bioavailable. This non-specific delivery. This, as I told you, these herbal drugs they give this total systemic effect. It controls whole system, then a specific part. So this non-specific delivery, poor bioavailability, that is also a problem. Then this administration of this high dose, sometimes or most of the cases it is high dose is required. This lack of this drug release profile, so not much of work also that this release profile is not defined or determined. Then these toxicity problems. So these are the problems in these novel drug delivery systems, or these are the problems can be overcome by these novel drug delivery systems. Now this nanotechnology, you need not to say that this is a multidisciplinary approach, and it has this wide applications of this in the novel drug delivery systems, and one of this nanotechnology is the nanoparticles or this nanoliposomes. Here the different type of nanoparticle systems are given. Everybody know about this one. This nanoparticle and nanoliposomes, a little bit differences are given, but I think everybody know where this. So details of this liposomal systems and nanoparticles, different type of nanoparticles are discussed here. So this advantages of nanoparticle also we can consider in respect of these herbal drugs. This particle size and the surface characteristics of nanoparticles, this is a very important aspect. This can be easily manipulated to achieve this both passive and active drug targeting. This nanoparticle can control the sustained release of the drug and during these transportations and the site of these localizations. This sustained release, how the drug will be released and how it can be controlled and how it can be targeted. So this can be possible through this nanoparticle also. This subsequent clearance of this drug so as to achieve this increase in this drug therapeutic efficacy and reduction of the side effects. This is one of these major advantages. The drug loading is relatively high, and this drug can be incorporated into this system, nanoparticle systems, without chemical interaction. That can be also possible. The site-specific drug targeting or drug targeting is possible with this ligand, and this system can be used for the various routes of administrations, including this oral or nasal or parenteral or other routes can be used. So these are the different advantages of nanoparticles which can be applied in these herbal drug delivery systems. Now this application of this novel drug delivery systems into this herbal formulation, in the recent years there are a lot of advances have been made, and you have seen this, a lot of papers have been published on this development of novel drug delivery systems for plant actives or extracts. These novel herbal formulations are like polymeric nanoparticles or nanocapsules or liposomes or phytosomes or nanoemulsions or microspheres. So these have been reported using these bioactive or plant extracts. Novel formulations are reported to have these remarkable advantages over these conventional formulations of plant actives. The extracts with this include this enhancement of the solubility, bioavailability, the protection against the toxicity, enhancement of this pharmacological activity, enhancement of the stability, or improved tissue macrophage distribution, and the sustained regulatory resistance, and many other things can be done on these herbal formulations. So, these major challenges which we can overcome for this herbal formulation is the solubility enhancement of this poorly aqueous soluble this hydrophobic plant extracts, better permeability through these biomembranes, the faster absorption and good bioavailability, the modification of biodistribution and pharmacokinetics, and this drug targeting, crossing biological barriers or the membranes to deliver these active components to specific organs, slow inhibition of drug metabolism, and increased gastric residence, toxicity, etc., etc. So, these are the challenges before these herbal drugs. Now, these challenges can be of course optimized by this properties. Already I told you this size of this nanoparticle or physicochemical properties or surface property, degree of biodegradation, biocompatibility, toxicity, and many other things. Now, these are those who are working on this nanotechnology, they are very much acquainted with this one. Now, there are some examples. This is how this nanotechnology used in these herbal drugs. This curcumin. Curcumin, you know that this is a very famous drug, traditional drugs, widely used everywhere. It's basically from this plant called this Curcuma longa. It is available in this most of these parts of this world, particularly Southeast Asia. It is used as antioxidant or anti-inflammatory or antiviral, many, even also in the cancer or diabetes or cardiovascular diseases, these curcumins are used. Now, these major problems associated with this curcumin is the solubility or the bioavailability and the stability. So, these three problems, as you know, this nanotechnology can solve easily these three problems. So, this curcumin is reported to be this unstable in the gut, and little amounts of this curcumin that pass through this gastrointestinal tract rapidly and are rapidly degraded. So, this nanotechnology has been successfully utilized here and been proven to be very effective in solving these problems. How? See, these are some papers. These are the plants. This in vitro therapeutic efficacy of this nanoformulated curcumin and the free curcumin against a panel of human pancreatic cancer cells lines showed this comparable results. Means this nanoformulated curcumin gives the better results than this the normal. Novel polymeric, this amphiphilic conjugates with this lipophilic and hydrophobic segments showed this minimal toxicity on these HeLa cells. These are the reported. This nanocrystal solid dispersions of the curcumin and amorphous solid dispersions and nano emulsion exhibited this marked improvement in the dissolution behavior when compared with this crystalline curcumin with a significant improvement in this pharmacokinetic behavior. This pharmacokinetic behavior of this nanocrystals or this nano emulsions has been much improved than this amorphous curcumin. So, this the nanoformulations, it has given this much improvement in this curcumin. Similarly, this Taxol or this paclitaxols, these are the very widely used drugs. It is a very potent this anti-cancer drug, particularly in the leukemia and other tumor, in the cancers. This Taxol suffers from this fact that it is water insoluble. Ethanol and chemophore are generally used for the solubilizations which have very side effect or toxic effects. But this polymeric nanoparticle drug delivery systems for paclitaxols improve the therapeutic index as well as this adverse effects of this drug, the chemophore is avoided. This is the advantage. So, this the nanoparticle strongly enhances the cytotoxic activity compared to this the taxols. This paclitaxols was loaded in sterically stabilize these biocompatible and biodegradable phospholipid nanomicelles, and it expressed more than this 10-fold higher this P glycoprotein than its the parental sensitive cell lines when it is compared with this MCFC, or it's compared with this paclitaxel resistance in the human breast cancer cell lines. So, this is the advantage it is found. Similarly, these other traditional medicines like this camptothecin, these PLG nanoparticles loaded with this camptothecin, it is conjugated with these antibodies against these colorectal tumor cells, and it was observed that this uptake of this antibody nanoparticles was increased in the cell compared to this nanoparticles without this antibody, and increase the cytotoxicity of camptothecin. So, camptothecin also it can be successfully used in this nanotechnology. Other very common drug, this ginkgo biloba. You know, ginseng is a very widely used for this purpose, as you know, everybody. But it has this, the drugs can be embedded and dissolved in nanoparticles, and it can be absorbed or coupled on this surface. This encapsulating drugs with these nanoparticles can improve this solubility and the pharmacokinetics of the drug, which is the major problems of this, the ginkgo biloba or the ginseng. The leaf extract of the ginkgo biloba has been widely used for brain cell activation properties. This nano-sized ginkgo biloba extract is expected to activate the brain cells and work on this treatment of the Alzheimer's disease, like the loss of memory and other things. So, in Alzheimer's disease, this nano formulation of this ginkgo biloba or ginseng is widely used. The leaf extract of this ginkgo biloba has been widely used for this brain cell activation properties. The nano-sized this extract is expected to activate the brain cells and work on this treatment of Alzheimer's disease. Other drugs like in diabetes or hypoglycemic drugs, this 2-prand, the traditional plant, is widely used for this diabetes or hypoglycemic effect. One is the Cesium, this Jambolena and Gymnema sylvestra. So, these have been shown to have relatively more hypoglycemic effects than their encapsulated these extracts. The nano formulations of this active, these phytoconstituents or gymnemic acids has shown this to improve their physiological action, pharmacokinetic and pharmacodynamic behavior when it is compared with this that type, same type of these marketed products. Licorice is another important traditional medicines used many parts of this world. This is glycyrrhizin and glycyrrhizin acid. These two are the main active component. If these they are used in the different diseases or the liver treatment also. So, the chitosan nanoparticles were prepared and modified with this the glycyrrhizin and confirmed that this nano encapsulated the drugs were preferentially accumulated in the rat hepatocytes by ligand receptor these interactions. It is also found that this cellular uptake of these liposomes modified with glycyrrhizinic acid by this rat hepatocyte was 3.3-fold higher than that of these unmodified ones. So, overall this pharmacokinetic behavior and physicochemical factors related with these drug delivery systems are considered to be the primarily responsible for the improved targeting and therapeutic effectiveness. Therefore, dealing with these factors during the development of these nano herbal formulations can lead to more promising treatment of for acute and the chronic disease. Therefore, this nano formulation can overcome the disadvantages of this poor aqueous solubility, physical instability, the low drug absorptions, lower bioavailability, and slow pharmacological actions, drug targeting, faster eliminations, toxicity, and all these problems of these herbal drugs can be overcome by this nano formulation. So, basically, so this in my conclusion, the traditional medicines will contribute to this human health care in the 21st century. It was helping from this I think 5000 BC from these earliest civilizations to till today, but today if we need to explore it. we need to modify its delivery systems so that it will be very useful. There are many challenges, of course, to this safety and effectiveness for the use of traditional medicines, and that I think in the different laboratory working, this overcome the challenges of these drug delivery systems. But there are huge scope for this nanotechnology based traditional medicines. That is the expectations. So we have a long way to go. We have to explore these traditional medicines in the different country. Particularly, it will improve a lot in our healthcare systems. So these are the different references where this herbal drugs or the plant extracts or nanoformulations are being published. Thank you very much.